Thank you, Jess, for inviting me. It's very exciting and slightly nerve-wracking to be back, but really fun to be back. And I've been talking to students this morning, and it's just great to hear about OPR and all that's happening here. So as Jess mentioned, I work on infectious diseases, and a lot of my work is trying to think about how infectious diseases spread in human populations. So if we think about an infected person being introduced into a population, shown in red, we think what happens through time, the infection might spread, we might see recovery, and this is kind of the type of data that we often see, typical outbreak curve, number of cases over time. And I try to really, in probably the last 5 or so years, I've been trying to think a lot about what are the different factors that drive these dynamics that we see in a population. So how quickly do pathogens spread? Where do they spread? What are the populations at risk? What time of the year do outbreaks happen? And there's many different factors that affect this. So demography is really key here. So births add to the pool of susceptible population that can be infected, whether it's a new pathogen or endemic pathogens. So births are really important. Mortality obviously is important for understanding the burden of disease, but also things like migration. So I do some work on thinking about where are people traveling to, what places are connected. If two places are more or less connected, how might we think about risk of pathogen transmission between these meta populations? And then kind of in the other realm, I think a lot about climate drivers and environmental drivers. So things like temperature, precipitation, humidity, how does that affect disease transmission? And then lastly, and probably what I'll show mostly today, is thinking about factors beyond things like birth rates and death rates, and beyond things like temperature and climate, but thinking about these other dimensions, social, economic, and political dimensions, which are often a lot harder to measure, but as social scientists and a lot of you working on this, there's a rich body of literature in the social sciences of how to kind of measure these factors. And so what we've been trying to do is think about, well, how do we measure these social, economic, and political factors that might affect disease dynamics? And then how do we put it into typical models that people use for studying infectious disease transmission? And so what I'll kind of talk about a little bit is how we think about what are the extrinsic and intrinsic drivers. And mostly today, I'll focus not on climate or the human demography side, kind of these social and political drivers. How do we statistically estimate it? Often the only data we have is something like this, number of cases. And so how do we, if we know many of these factors are at play at the same time, how do we statistically estimate these relationships? How do we then mechanistically model them? So this is a lot of what sort of people who are building formal models work with. They're working with mathematical models that mimic how the biology of virus and the transmission process. And so how do we think about modifying these types of models to incorporate some of these other factors, and then what that might mean for the future burden of diseases as these factors are changing. Okay, so today's talk, I'll talk about some work that I've been doing with colleagues at Berkeley, thinking about how do we model heterogeneities in disease transmission using data from surveys. So we're using very simple survey methods and survey tools to collect information about people. What we're really interested in is thinking about how do diseases spread in populations. And so we've been thinking about how do we conduct surveys to measure things like maybe the age of the person, so we care about simple demographics, so age, gender, but also some things about behavior. How many people are our survey respondents connected to? What do their networks look like? Who's vaccinated, who's not, and things like that. And then once we're able to kind of get a grasp on how these different factors vary in our population, we can then try and put them into these mathematical transmission models to think about how does that shape the disease burden. I'm also happy to take questions, so feel free to interrupt as I'm going along. Okay. So when we think about these types of models, I typically, almost everyone in this room has seen some version of this, when we think about these susceptible, infected, recovered, these SIR models, where we have the population divided into body compartments if you have a susceptible pool. Birds can add to this. Migration from another population can add to this. You can recover from a pathogen and move back if there's waning immunity in a susceptible compartment. And then there's a transmission process whereby if you encounter infected people, a susceptible person can become infected, and then after a while, they can recover. And when we think about this process, I think ecologists and epidemiologists have long recognized that this is an inherently social process. We need people to come into contact with each other in some form in order for the transmission to occur. So whether we're thinking about sexually transmitted diseases and sexual networks, or we're thinking about childhood diseases and how children are coming into contact, there's inherently some social structure that's determining how people are coming into contact with each other. But these kinds of social and behavioral aspects are hard to measure, and social scientists kind of devote our whole lives to thinking about how do we measure these things. And what we're really trying to do here is find a way to measure these things and maybe find a way to incorporate them into these kinds of mathematical models to add a little bit of nuance to what we think the object dynamics would look like. Okay, and I'm going to talk a lot about something called the contact rate. So I just wanted to give you a sense of why the contact rate is important. So when we think about the R0, this is the basic reproduction of the disease, or for demographers, this is the NRR. How many new infections would you have in a fully susceptible population if you introduce one infected person? So R0 greater than 1, you'll see an outbreak. R0 less than 1, the infection will die out. It works exactly, the math works exactly the same way as if you would calculate NRR. But there's a few different, and this is a very simplified version of R0, but if we think about this very simple process, there's people coming into contact with each other, and this is determined by some sort of social, political, and environmental structure. That contact rate will, if you come into contact with an infected person, there might be some probability of infection given contact that's going to determine whether or not you get infected. And then the duration of infectiousness matters for R0 as well. So the longer duration there is, more people that can be infected in a fully susceptible population. So this contact rate is really important, and it shows up even in this very simple SIR models. We care a lot about how people are coming into contact with each other and what rate are they coming into contact. And I should say one other thing. In a typical SIR model, we're going to assume that the population is really well mixed. So this is the kind of mass action principle. You can imagine people moving around like gas molecules, and your probability of running into an infected person is just a fraction of the population that's infected. So very well mixed homogeneous population. And the way that modelers have kind of thought about how to incorporate heterogeneity in the simple framework is to keep it still quite simple, but think about subdividing our population. So if we now think that there's two different groups that might have different kinds of behavior or demographic characteristics that put them at different levels of risk, or maybe they have different kinds of contact behavior, we might want to split that population up. So age is a sort of classic way in which ecologists and epidemiologists have subdivided the population. Age goes a long way in capturing many of the behavioral things we see over the life course. So contact rates, I'll show you, varies a lot by age, but other factors as well. So your risk of dying from an infectious cause also varies with age. And so it makes a lot of sense to divide the population by age. And when we do this, we keep the model still quite simple, except now we have two groups, they interact with each other. And so this interaction is really what me and my colleagues at Berkeley have been trying to spend a lot of time trying to measure. How do people in different demographic groups or different groups defined by some other measures, such as political identity, come into contact with each other? And one of the classic studies to collect this kind of data is the Palma study that was done by Joe Masong and colleagues in 2008, where they collected data on how people of different age groups come into contact. So this is a contact matrix that you basically would put into this kind of model, right? You need a matrix to tell you what's your within and between group contact. So they had people keep diaries. So this is age of their participants who kept diaries in their study, and then these people who wrote down in their diaries what was the age of the people they were coming into contact with on a daily basis. And what's plotted here is the average contact rate, and so that lighter or the brighter colors are higher average daily contact rate, and you can see most people mix with people in their own age group. So there's a lot of homophily in age contacts. And then you can kind of see these whiskers of the side where people are coming into contact, parents with children, a little bit of grandparents with grandkids, and so on. And so this is the type of data that modelers are interested in putting into their age structure model to kind of predict what algorithm dynamics would look like. And this typical pattern, and this kind of study has been replicated in many settings, and this very strong assortative pattern actually shows up in many contexts. So here's one example of data by one of my students in Bangladesh who collected this data in a very rural area in Bangladesh, very different population, really high average context. So the diagonal people are having 24 contacts on average per day, whereas the polymod study found something between 8 to 12 average daily contacts. So very different numbers, but you kind of still see the same assortative pattern.
So just clarification.
Yeah.
How do you define contact in this?
Yeah, great question. So in the studies I will show you, and also in the original polymod study, contact is defined as either a conversational or physical contact with a person. In our study, which I'll show in just a second, we define a distance as well for the conversation because we were interested in COVID transmission and kind of heard a little bit about distance. What this doesn't capture is contact that is, so suppose you went to a grocery store and there's lots of people around you, but you only talk to 3 people. Those 3 would be captured in our context. In this Bangladesh study, we actually tried to capture things like if you went to a grocery store or a public market or a mosque in the Bangladesh context, or public transport.
Numbers.
A little bit. This data is actually taking out those contacts and only showing the physical and conversational. People are just having a lot of contact. And the Bangladesh context is really interesting because males and females have very different types of contact matrices in Bangladesh, which we don't really see as much in the US or in the polymod study. This is very much a work in progress. So I don't have other results from Bangladesh, but lots of interesting. Okay, so this polymod data has been really phenomenal for the field, and the thousands and thousands of modeling papers have been published using this polymod data to parameterize it. But unfortunately, data for the US had not really been collected. And when the pandemic hit, when COVID-19 hit, one of the things that my colleague Dennis Feehan, also a graduate of this program, I think this is Dennis's photo in the brick wall downstairs from when he was here. So make friends with your peers in your program because they might become your lifelong collaborators. So Dennis is my colleague at Berkeley now, and Dennis and I were, when the pandemic hit and the lockdown had been announced, we were trying to figure out what could we do. And Dennis is a network scientist, and we've done a lot of great work on networks, and so we decided to kind of join forces and say, okay, well, maybe the best thing we could do is just collect some polymod-like data for the US, and maybe we'll learn something about how the US is different and also how the pandemic is a totally different context. So the early modeling papers from the first year of the COVID-19 was still using polymod data, which to us felt a little bit problematic because polymod data is collected 2008, a while back, and also not from the US context. And so this was the motivation for starting the Berkeley Interventional Context Study. So we started collecting data on context. We asked people about physical or conversational context, but we also asked about a whole host of other types of behaviors. So we asked about if a vaccine does become available, would you accept the vaccine? We asked about we had a special module on occupation because we were really interested in who's able to shelter at home and who is not. So we asked about essential work. We asked about masking, whether or not people felt safe, like perceived risk, and many other things. And then we repeated the survey over the first year and a half of the pandemic. So our first wave was the week after the lockdown was announced in the Bay Area and also a few other parts of the US. They were all not at the same time, but our first wave is roughly when the lockdowns are starting. And so today I'll talk about some of the results from the BIX project, and then we've done some follow-up surveys in Illinois asking similar questions, but now also expanding this to think about not just COVID, but other respiratory pathogens. So we asked about flu vaccine and so on. So this is what the timeline of our data looks like. We had a national sample that's roughly around 1,000, and then we had city-specific samples. The cities were not chosen with any great thought other than we lived in the area, so we were interested. We had a colleague, Megan Todd, also a graduate of this program, who was at the Philadelphia Department of Public Health and was collaborating with us. And Megan asked for some data, so we collected it. We had some colleagues in Boston as well who were interested, and then we kind of chose Atlanta and Phoenix as the cities that were different, but for no really systematic reason. Dennis and I were really scrambling to put this together, and one of the results of this is that what we ended up using is an online panel, which has many problems that we knew at the time and have made a lot of effort to correct for it, and I'm happy to talk about it. But the things I'll say about it is this is not a sort of typical address-based sampling where you would, random sampling that you do for typical surveys in social science. This is an online panel where people opt in to answer these questions. But it's cheap to do. We could do it really fast. And so because we were doing it, we wanted to deploy the survey the next week. We needed something that was quick and cheap. One other thing I will say is that Dennis and I are now with some CDC funding running. So, and with colleagues, Audrey Dorellian, also a graduate of this program, we're running some surveys where we're doing this classic address-based sampling and these online panels, asking the same question to see if we can leverage online panels to tell us basically what we know with some correction, given that we'll have ground-ish data. And then this is quota sampling, so it's done so the demographics of the panel look roughly like the population of the places we're interested in. So we sample for the cities and then also the national sample. And then we use these, survey weights and so on to try and account for this, but I'm happy to talk more about online panels. I think especially in the age of AI, online panels are becoming really problematic, but it was a really great tool for COVID because there's lots of really great data being collected very quickly that you could do. Okay. So this is the type of data we collected. This is the same format that I was showing for Polymod, where we had the age of our participant and the reported age of their contacts. And you can see that contacts are really low. So Polymod was something like 8 to 12. And this is just when the lockdown is announced. And so people are barely having any contacts. This is roughly like the average household size. So mostly people are having contacts within their household. And then Dennis had some really great data from beforehand about networks that was not quite the sort of disease relevant contact networks, but close enough that we said, okay, maybe we can compare to some baseline data. And so this is from a totally different study, and we're looking at the difference from our collected data and this previous study. And you can see the biggest drop in contacts, so this is showing the difference. The largest drop is for these younger age groups. So these age groups experience, you can also see as well. The biggest drop in average daily contacts was for these groups.
Ayesha,
Yeah.
Did you look at how, what we know in fact for work, you did this really varies by socioeconomic status. And I imagine people who are able to respond to identify something online and respond to something online might be from one group as opposed to kind of a sense of what's happening more broadly. So do you have a sense of like
the sample and who they are? Yeah. And we did okay, so our education and income sample, because it's a quota sample, they roughly do look like the US population. Even then, I would think there is some selection bias into who is in these panels. I'll show you some results in a second where we looked at this essential worker, non-essential worker. We had a fair share or enough to at least statistically estimate differences between essential workers and non. But I do think that the online panel is probably still biased for those reasons, yeah. So this is our data going forward in time. So this is going all the way up to September, and you can see that already the sort of people are moving back up to where they were pre-pandemic. And so there's this dramatic drop in contacts early on in the lockdown, but then very quickly we started to see contacts go back up. And we can estimate what this means for R naught. And so this is what Dennis and I tried to do in our first paper, is really take this data and use it to tell us something about what is the hypothetical R naught in this population. And so wave 0 R naught is below 1 with our estimated contact matrices. And this is just, looking at what's happening over time. So ignore the colors, it's complication. That's not really important. They tell the same story. But we can see that the pretty pretty quickly, by the time we get to even wave 3, the contacts are not quite close to what it used to be before, but pretty substantial increase, and we see that reflected in the R naught as well. And then we can look at how these contact rates vary by different characteristics. So these are results from a pretty simple binomial regression model. And we can see there's not really a difference between weekday and weekend because most people were working from home. We do see some interesting differences by race, ethnicity. I have a student now who's kind of working on these race and age structured contact matrices to try and understand how much of the variation in mortality burden could be explained by differences in exposure and contact rates. I think it seems like not a lot. Contact rates were different between racial ethnic groups, but not to the scale that it would explain the massive sort of disproportionate burden that Blacks and Hispanics in the US have. We can also look by age. So in our survey, males had roughly higher contact and young males had the highest number of contacts. And then there's some city specific variation as well that we would, thought were interesting, but mostly what we thought is like places look more similar than different. And so green is wave 0, pink is wave 1 over time in all these cities contacts. Well, slightly different patterns, but they're going up. Okay, so we see that there's a fair amount of difference in contact patterns over time. And so we wanted to ask the question of, okay, well, are there substantial spatial heterogeneities if we look across the US? The city samples didn't seem to give much of an answer, but cities are unique. And so we wanted to think about, well, what do different states, for example, look like? And then what are some other factors other than age? So age is important, but what are some other things that might be important for explaining differences in contact rates? Here. So Casey Green, who was a PhD student with us at the time, really fantastic work on trying to come up with a method for taking these national level contact data where we really don't have the resources to go into every state and carefully sample, but maybe we can take this national level data, learn something from the covariates that we've collected information on. So this is where we fit on these Bayesian hierarchical models, and then post-stratify so that we can match what the demographics of different states look like to tell us something about patterns. And so Casey did some really great work looking at this both in one single time point. So this is July 2020, and I'll show you in a second over time. And you can kind of see this sort of assortative pattern looks pretty similar across the state. That age signal is really strong. And so even though the other covariates matter, this age signal kind of shows up. You kind of see this diagonal light up in all places. But there are some differences. So there's just pulling out 2 examples, South Dakota and Washington. So Seattle was really hard hit by the first wave of COVID-19. And you can see that sort of reflected in I kind of looked at perceived risk as well, but you can see that the contacts are pretty low even in July, whereas South Dakota, you can see so it's an average of about 4 contacts per day, which is still low, but higher than we saw in other parts of the country. And then we can look at how this changes over time. So this is just looking at those contact matrices, looking at not quite R naught, but something that's correlated with R naught. We're looking at the dominant eigenvalue of these matrices over time. One would be sort of if you went to your baseline level. And if I, again, pull out the same states, you can see that Washington state remains really low. So this would correspond to a pretty low R naught, whereas South Dakota, you can see it jumps up. Here's a big wave. People are worried. They start social distancing again. It goes down. And then you can kind of see this trajectory. Okay, and then we asked the question of what are other factors that might be important. And so maybe Yonatan's question here about thinking about demographic and socioeconomic factors. So Ethan, who was a PhD student with us, asked this question of how do these contact rates vary by occupation? And so there's people in our sample who report working from home. There's people in our sample who report going into work every day. There's a whole range of types of work scenarios in our sample, but we did a lot of work to classify people into what we're calling these sort of high contact occupations. These end up, if you look at the categories, mostly essential workers. versus adults in what we call low contact occupations. And there's a substantial difference. So this is a time period where contacts are really low across the board. People are having on average, maybe 3 or 4 contacts, but you can see that adults in these high contact work were having on average about 8 contacts, which is almost the polymorph level. So this is a group that were not really able to social distance themselves in the same way at all. And we can put this into a model that's actually still a very simple SCIR model. We just now added a few more compartments to show this heterogeneity and risk based on your occupation. And we can ask questions like, well, if we have a vaccine, so this is, we were writing this paper before there was a vaccine, but we can ask the question, if you have a vaccine that we think is going to directly protect those who are at highest risk, we definitely want to vaccinate the elderly and you know, maybe we could be targeting at nursing homes and so on and hospitals. But maybe there's some indirect protection that we could get from the vaccine. So if you vaccinate the people who are having the highest number of contacts, they may indirectly protect people in the highest risk group. So we tried a whole range of vaccine scenarios because we didn't quite know what were the properties of the vaccine. And so the bottom line that we found is like, it always makes sense to prioritize the elderly because the age gradient of mortality for COVID-19 is just so stark, even with it washes out the indirect effects. But if you think about sort of the burden that hospitals might face, so what's the burden of clinical infections, it actually makes sense to then prioritize those who are in these really high contact occupations. And so if you're able to have a strategy where we can identify them, that's and then in the US, actually, the strategy was a little bit like that. There was the priority was for the elderly, but also school teachers, people who worked in nursing facilities, people who worked in some of these essential works were actually prioritized as well. Okay, this is really the last set of results, and then I'll wrap up. So this is work by Chris Surya, who is a PhD student with us right now. And Chris was really interested in a question about, okay, well, what else might explain heterogeneity in behavior? We see variation across state, you know, age is important, but what else is going on? And so Chris made this figure looking at 3 different measures. So we have total number of contacts, so that's just the degree of your network. What percentage of your contacts are the people who wear a mask? And then what percentage of your contacts are, or what percentage of this population that we survey say that they would get a COVID-19 vaccine. And we can look at this across several characteristics. So these are showing the differences. So the first bar is showing the difference between under 65 and over 65. You can see that age explains almost a lot of the variation that we see in contact rates. But these other factors need to kind of matter a little bit as well. And the second most important factor in our data that explains differences across groups seems to be whether or not someone identifies as a Democrat or a Republican or an independent. So in our sample, we see that Republicans have higher number of contacts. They're less likely to have contacts with people who use, less likely to be in a contact where a mask is used by either party. And they're less likely to report either saying they would get the vaccine, or in our last wave, we can measure whether or not people actually got the vaccine. And Chris is really sort of was interested in this question of, okay, well, could some of the difference that we see in mortality by whether or not a county is Republican leaning or Democratic leaning be explained by behaviors? There's many things going on. So policies are different. The people who live in these, the age of people in these counties are very different, but we were kind of interested in trying to tease apart how much of it could be due to behavior. So this is looking at COVID-19 mortality by county partisan composition. You can see at the start, Democrat-leaning places are much harder hit. So this is New York, Seattle, all the cities getting hit. And then you can kind of see the split happen, and Republican-leaning counties have much higher daily deaths over time. And then in each of the 2 subsequent waves, we see the Republican-leaning counties have a higher wave. So many things are going on. So age is a key thing that's, I think, different between people who identify as Democrats and Republicans, although that is now less clear how important age is. But so this is showing some results where we control for every observable thing we can in our sample. So this is controlling for age, controlling for policies that, you know, whether schools are open, whether the governor of your, this is at the county level. So I think we have county level policies. There's a really great data set on this that if anyone is interested, I can point you that Chris found. So we control for policies, age, gender, the COVID-19 mortality rate in the month prior and the 10 days prior and all sorts of things. And we still see this difference between Republicans and Democrats persist. So some of it is explained away by things like age, but we still see this pretty big difference between in number of contacts reported by Republicans and Democrats, in mask usage, and then in probability of vaccination. And the interestingly, we also see if we look at counties, if we look at Democrats living in Republican-leaning counties, we see them behave a little bit like the Republicans. When we see the Republicans in Democratic-leaning counties, they behave a little bit like Democrats. So clearly something is going on. There's a selection story. There's probably things about policy that we're not capturing. But just with all the things that we can control for, we still see this sort of persistent difference by political party. And so we asked the question, can we put this into a mathematical model to see what this might mean for outbreak data? So still also in the same simple SIR framework, people going from susceptible to infected to recovered, we have waning immunity now. But to explore the impact of masking, we now split our population into groups that don't wear a mask and groups that do wear a mask. I won't show the vaccination picture because it makes it much more complicated, but you can imagine we have we can have vaccination here as well. And then we have two processes. There's movement between unprotected and protected groups, so you can move into mask wearing status, and then that wanes over time. We see this in our data, and so you can move back into unprotected status. And then we run this sort of transmission model for Republicans and Democrats and Independents. We have three parties in our group, and then each of them kind of follow this transmission process. And we can think about the sort of force of infection for any particular group is really going to depend on this contact rate. So how much mixing is there between your own group versus mixing in with people who are identified as a different party. So I'll talk about them. So this is kind of what happens if we just simulate a hypothetical outbreak. So we're not, again, I think we're trying to not, we're not, trying to exactly predict what happened during the first year of the pandemic. We're saying if we take the observed data in our survey, we put that into a mathematical model, what does that say about a hypothetical outbreak in this population? So we see that in our simulations, Republicans do have higher cumulative incidence, Independents are somewhere in the middle. The Republican peak happens a lot sooner, which makes sense. They have higher number of contacts, and so we see the Republican peak happen first. And then the sort of time to peak incidence in both the peak are higher for Republicans compared to. And this is kind of what those simulations look like. So the dashed orange line is just taking the observed behaviors of of Republicans, Independents, and Democrats in our survey and putting it into the model and simulating an outbreak. And then the green one is a sort of layer, adding on a layer of another scenario on top, which is saying, okay, if we pretend that everyone in our sample now behaves like the most cautious group, which turns out to be the Democrats in our survey, but if they behave like the most cautious group, how different would that outbreak look? And it does look pretty different. The burden is shifted down quite a bit. If every group is behaving like Democrat, we see the peak happening a lot later as well. And then the sort of key piece of this is homophily. And it turns out homophily is really hard to measure in these kinds of surveys. So homophily is telling me how much are Republicans mixing with themselves versus with others. So we can ask people in these surveys to guess about age, race, ethnicity, gender, and so on. It's really hard to ask people to guess about people's political affiliation because these are encounters like you talk to someone at the grocery store or have a chance encounter on the street. And so it's, for most of these contacts, we might not know. Although we're trying to do some work to estimate this, which seems to be hard. But we're in the simulation, we can simulate it to see, well, how important would homophily be? And it turns out homophily is really important. So this thing that we can't measure, unfortunately, turns out to be really important. So our data is a way for us to model homophily. So you can think of it as a homophily parameter. As we move from left to right on the x-axis, we're having more contacts within a group. So homophily is increasing. So this is Republicans having more contact with themselves. And then we also vary what fraction of our population is Republican, because different counties have different compositions in the US. And you can see that homophily is bad for the risky group, if Republicans have most of their contacts with themselves, the outbreak spreads a lot faster and they are at higher burden and higher risk. And that risk also goes up with the fraction of the population that's Republican. For Democrats, mixing with their own group is protective. So they have fewer of their contacts with Republicans who have larger networks, so their degrees are larger, and therefore they're at more risk. They're also less likely to wear masks or get vaccinated. And so among these protective for Democrats, but as the fraction of Republicans increase, we see more cases in the population. And then this is just looking at. I said I would stop with that. I'll show one other result from our new surveys, and then I will really stop. Okay, so this is by my postdoc Soren, who took these surveys that we did during COVID time and said, okay, well, what's happening today? And one of the things we're really interested in today is vaccination behavior, because we think that's going to probably determine a lot of what the dynamics might look like going forward. And so we asked this question not just for COVID, but for flu, to try and get a sense of how people are making decisions about COVID and flu. And you can see that age, again, biggest prediction, not just with contact, but also vaccination behavior. Age turns out to be really, so demographers, you have probably thought about this and known this for a long time, but in this context, also age turns out to be really important for whether or not you are getting a COVID-19 booster or a flu vaccine every year. So there's income in the form education seems slightly important, but again, political affiliation shows up as quite important. We see significance. So Democrats are much more likely to get either of the vaccines. What's really interesting is when we dug down into these results and we looked at what are the reasons people say for getting a vaccine versus not getting a vaccine. So it looks like, you know, people who get a vaccine all kind of look the same. They kind of say they're doing it to protect themselves or others, and people who don't get a vaccine are all, you know, very different in their kind of own ways. And so, and it differs a lot for COVID and flu. So the reasons people don't give for not getting the vaccine seem to be pretty different for COVID and flu. And so for COVID, it looks like a lot of the reasons are kind of distrust of the vaccine. For flu, it seems a lot of people say, you know, they're just not worried, they couldn't be bothered, or lack of time. Lack of resources. So it seems to be something at play between, you know, kind of people's socioeconomic conditions, demographics, political beliefs, and how they're behaving for these two different flus. What's also interesting is people make different choices for themselves versus their children. This is the same data, and we can look at within household what's happening. And we can see that we have lots of households where only the children are vaccinated, adults are not getting vaccinated. We also have a fair share of households where the adults get vaccinated, but they don't vaccinate their children. So we're kind of doing some follow-up surveys to figure out how are people making decisions for themselves versus their children. But this turns out to be really important because COVID-19 vaccine eligibility, as you heard, is kind of changing in the US. And so we took this data and asked the question, if we made children ineligible for the vaccine, so health insurance doesn't cover it anymore, what impact would that have for the population? And it turns out the biggest impact is actually for the elderly. And so if we take our contact data, we know that children mix a lot and they have a lot of contacts with the elderly in the population. Taking away the vaccine eligibility for children actually has the biggest impact on the elderly in our population. Again, this depends on the extent to which we think that there is really good indirect protection versus not. So this I think there's some nuance here, but we try a whole range of scenarios. And even in pretty conservative scenarios, reducing vaccine eligibility for children can have sort of these indirect impacts on populations that are at risk, especially in light of, you know, there are these household structures where we see the children and adults are kind of choosing to have different, children are not choosing, but adults are choosing to have different kinds of decisions for themselves than children. I think this is my last slide. Okay, so I think hopefully I convinced you that this kind of data is important. I think we should be thinking about how to collect these kinds of data in, I think, more clever ways, really trying to understand things about the network property and who is coming into contact with whom. I think there's a huge, rich body of literature in doing this for sexually transmitted diseases and less so for respiratory pathogens, but we think there's probably some importance in thinking about this for respiratory pathogens as well. So age differences are large. You can see that, but we also think these differences by other dimensions like occupation, party affiliation are also quite important and have relevance not just for the magnitude of an outbreak or the timing, but actual policy decisions that are okay. And with that, I'll say thank you. We have all our code and all our data is available. So if anyone wants to, there's tons in there that Jonathan and I just haven't had time to dig into, but if folks are interested, it's all there.